Hi, thank you. It's great to be here, and I'm excited to tell you a bit about the work we do in our department. So first of all, here is a nice view from our rooftop terrace. So Tübingen is very beautiful and hilly, and fortunately our institute is at the top of one of these hills. So we have a great view there. So our group is relatively new at the Max Planck Institute. So we just moved in 2016, and before that we were at Cornell University in upstate New York, where Ruth had first started her own research group there in 2008. So Tübingen, if you're not familiar with this, is, I don't know if that's very accurate, but it's sort of the southwest corner of Germany. We're a little bit south from Stuttgart, and our Max Planck campus has a total of 3 institutes. So I'm at the Institute for Developmental Biology, but we also have cybernetics and intelligent systems. So technically there is the Friedrich Mischer Laboratory, but in reality this is just a part of the Institute for Developmental Biology. So our institute has a total of 6 directors. Some of you may think I can't count because there are 7 faces up here. It felt like it would be a disservice not to put Christiana Nusslein-Volhard up here. So she retired a few years ago, which is when Ruth took that director position, but she does still have an active research group at the campus. One of the things, though, is that right now we're really in a phase with a lot of turnover. So in principle, we really have 4 directors at the time, currently, excuse me. Unfortunately, Elisa Izaralda passed away unexpectedly last year, and Gerd Jurgens is retiring, so we're actively searching for 2 new directors. And the Institute is sort of changing a research focus, so we're really moving more towards host microbe interactions, as that is a common theme of some of these other research groups, and given our name of microbiome science, it's probably not surprising that we're interested in that as well. At the moment, we have a total of 7 independent group leaders. These top 4 are part of the FML, and we currently have 3. So again, a lot of turnover even with the group leaders. So we had 4 that just ended their stay last year and the year before, so I anticipate that we'll probably have a few more joining us soon as well. So a little bit about us, Department of Microbiome Science. So first of all, in case you are not familiar with the microbiome, to make this very simple, the microbiome are the microbes that live on us, so that could sort of be on the skin, and within us, so my favorite part is within the gut, which I'll tell you more about. So the microbes, in the microbiome field, we have a tendency to focus more on bacteria and archaea for the ease of cataloging these microbes, but this would also include eukaryotic organisms, viruses as well. So there's a couple things that I would want you to take away about the microbiome. One is that we really are heavily dominated by microbes. So the estimates vary, but for every cell on our body that is ours, that is a eukaryote, there's at least 1, if not up to 10 times as many microbes, suggesting that we're just as much microbe, if not more microbe than human. But what's important about that is also the gene content that they provide for us, because this really influences our health, in many positive ways. So the genetic content is about 10 to 100 times the amount of what our own genome encodes. And one of the principal functions that comes to mind, for example, is the ability to help us digest a lot of the complex polysaccharides in the diet that we don't have the enzymatic capabilities to do so with our own genome. So although today I'll be telling you more about host genetics in the microbiome, we really do have a wide array of research projects within the lab. So a few examples are the gut virome. We also look at bacterially produced sphingolipids and how this influences skin development, brain development. We don't just do humans and gut, we have maize and Pseudomonas. We have this really neat organism that seems to be able to live in seawater as well as the gut of polar bears. Diet and evolution, we're also very interested in methanogenic archaea. But unfortunately, I don't have the time to tell you about all of these things. So I'll give you a couple examples of what we do in the lab. So one of the things is we know that the gut microbiome does directly impact host phenotype in a number of ways. So the example I'm showing you here is with obesity, which is one of the most robust associations with the microbiome and human health. I would say inflammatory bowel disease is arguably about at the same level of importance. So the gut microbiome can influence host metabolism, so this can contribute to insulin resistance, metabolic syndrome, and this is caused by the microbiome through inflammation and basically the metabolites that can be produced by these microbes in the gut. So conditions such as obesity also have a very strong genetic component to them as well. And another thing a question that we had in the lab was sort of what influences the microbiome, and so who we have in our gut, and basically the relative abundance or how many of them are relatively there. So one of the questions that we had a few years ago was, well, how does the human genotype affect the microbiome, which we know impacts host phenotype? So this was a study of heritability, which is not to be confused with inheritance, which can be vertical transmission, but this is more a genetic predisposition. So these are usually done with large twin-based cohorts, and so we did this with a number of twins from the UK. Cohort name is Twins UK, not too unique there. But so one question, so oftentimes people will say height runs in families. So often, if you were to take a twin, if one twin is tall, the other one is likely to be pretty tall as well, because there's a strong genetic component to that. But one question was, so we could look at the microbiome as a quantitative trait, and ask that same question as well. And so not only the microbiome in terms of composition, but also the components or sort of function that are coming with this as well. So heritability is the proportion of variance in a quantitative trait that is attributed to host genetics. So unfortunately, I'm glossing over a lot of results here, but one of the things, so I said we had the question, how is the microbiome influenced, and what members of the microbiome are more or less influenced by host genetics? And this family Christensen-Alaceae was the most highly heritable, meaning that about 40% of the relative abundance of this bacterial family in the human gut can be attributed to host genetics. And one of the things that we also had some information, so we had BMI, or body mass index, information on these twins, and we saw that lean, healthy BMI individuals had more Christensen-Alaceae compared to obese. So that, and again, with some other results that I don't have the time to tell you about today, we wanted to ask the question, is this just an association, or could there be a direct causal effect? So one of the ways that we can study the direct effect of microbes on the host is through fecal microbiome transplants. So maybe you've seen this in the news, for example. So this is done with humans. Oftentimes this is done for the treatment of Clostridium difficile infections, which are usually a consequence of inflammatory bowel disease. This isn't just any sort of normal diarrhea. It is really horrible. It is extremely difficult to treat and may not always respond to antibiotics. And often antibiotics are what caused this in the first place. So basically there's multiple ways one could do the fecal microbiome transplant in humans. But what's pretty great is there is a very high success rate with this. And we can also leverage this same method in a mouse model to study the microbiome as a direct effect. So as I said, one of the questions I had was I wanted to see, well, does this Christensen-Alaceae family promote leanness? And so one of the things is that with obesity and the microbiome is we know, so I told you before that the gut microbes do directly influence phenotype, especially with respect to obesity. So we know this, not just us, but other research groups have done this as well, is that if you take a germ-free mouse, so these are mice that are 100% sterile, they're raised in bubbles, and if we take these same mice, they're reared in the same lab environment, they're eating the same diet, so it's a very well-controlled environment, and if we take stool from an obese human donor, it sounds gross, but mice love eating feces on their own anyway, so they do this gladly on their own accord. These recipient mice will have an increase in body fat accumulation, and sometimes we can also transfer impaired glucose metabolism and dyslipidemia to these mice as well, simply through the microbes from an obese donor. Conversely, mice receiving stool from lean hosts often stay nice and lean and a normal weight for a mouse at that given age and strain. So the question I had, so with this family Christensen-Alaceae, so I did an experiment where having obese donors that had very low levels of this family, so I predicted that obese donor stool would result in a heavier mouse. So I wanted to ask the question, what happens if I add in Christensenalaceae? So at the time, I was very lucky. There happened to be one cultured representative from this family. So I ordered this from the DSMZ, added this to this exact same obese donor stool to ask the question, if I add in Christensenella minuta, will I receive a leaner mouse? The answer to that was yes. So I was very careful to treat this entire community so they were getting the exact same treatment, except with the exception of having added Christensenella minuta. And so this is 3 weeks after inoculation. As you can see, the mice receiving Christensenella gained less weight, and the body fat percentage was also less in this case. So this is what I've been working on in lab, and this is my very large research group at the moment. So since I'm relatively new at the Institute right now, it is just myself, and I have one PhD student. And so the focus of my work is really following up on this. So the questions I have are, why are these mice putting on less body fat with this treatment? So what's going on in the host? How does this addition of Christensenella minuta affect the rest of the ecology of the gut microbiota in these mice? And then what does Christensenella do? Because there's very little out there that's known about this bacterium, and it's because it's a relatively new family. So as much as I would love to tell you all about my research, I figured I should give a broader representation of the department. So also on the theme of host genetics, so one of the questions, so I told you that Christensenalaceae is a very highly heritable family, but what gene or genes are driving this association? And the exciting answer to that is, I don't know. So we can assess, so we can do genome-wide association studies where we look at single nucleotide polymorphisms and associate them with health, or in this case, we wanted to look at this with the microbiome as a trait. So if any of you have ever sent off a saliva sample to 23andMe, this is basically the same information that they're taking to tell you if you have a genetic predisposition to a certain disease state. So unfortunately, just because we have to make so many statistical comparisons when there's so many SNPs, that we just don't have the sample size to have the power to detect which gene or genes are influencing Christensenalaceae. But one thing that has come out of these genome-wide association studies, not just from our group, but from others as well, has been an association with lactase persistence in bifidobacterium. So lactase persistence is associated with lactose tolerance, and this has a genetic component, and it varies geographically. So some populations have very high levels of lactase persistence. So this is basically, lactase is the enzyme that digests lactose. And so all of us, being mammals, have the ability to digest lactose at a very early age, but for some people, the ability to do that is lost over time. So basically what happens if you persist, the lactose, is broken down by lactase, and the host, if you're a lactose tolerant person, gets a bit of energy from that lactose. Conversely, if you're lactose intolerant, you don't have the enzyme, or making this a little simple, but basically the lactose can go through the gut, it's utilized by microbes in the gut, and this causes gas production, which is then what causes a lot of the symptoms of lactose intolerance. So if you're someone that is lactose intolerant and you're taking Lactaid, this is really just the enzyme lactase. So bifidobacterium, which this name may sound familiar to you if you're not a microbiologist, so this is something that is often used in probiotics. It's a type of bacteria that is used in a lot of yogurts, and this is increased with lactose intolerance. So as I just said, what we suspect is that because if you are someone that doesn't have the lactase yourself, it's going through the gut, bifidobacterium loves it, and then again, this is what can result in these symptoms. So one of the things is that, so I'll tell you about a study that we're doing in the lab, and of course just asking someone, how do you feel after you've had lactose, is not the best question because that's very subjective. But we can analyze this quantitatively by looking at two things. So the first is the blood glucose response. So this is sort of assaying the host response to lactose. I should also say too that in this case when we're assaying this, a person is fasting overnight, about 12 hours, and then on an empty stomach, just eating pure lactose, because stomach emptying and things like that can also influence the glucose response. So as I just said, so if you don't have the lactase enzyme, you won't have a blood glucose spike because you're not digesting it yourself. It goes through in the gut. And sorry, you probably can't see this picture. In case anyone is wondering, how are we measuring gas output? This is through the breath. And so we're measuring hydrogen and methane production. And conversely, someone that is lactose tolerant, they do have a spike. Sometimes it's mild to modest, depending on the functional capacity of their lactase. And then usually they have no gas production. So one of the things is that, there's a few people in our lab working on this project, and this was in collaboration with a lot of other places. So this is before I showed you the cute, sad cheese map of lactose tolerance. So here's a more quantitative one. And so basically this is a proportion of lactase persistence within a population. So we had 3 study sites. So the first here was in Germany, where lactase persistence is pretty high. Most people are tolerant. There was a study site in Vietnam where it is relatively low, and then in Gabon, which had an intermediate effect. And so at the site, Victor and Liam, as well as with local researchers at these study sites, they had about 200 mother-infant pairs. They did a wide array of characterization. So they also did the genetic testing to see what their genotype would be, these blood glucose and gas measurements, blood measurements, and many other things as well. So one of the things that's been really interesting, and not surprisingly, so we had some people that are lactose tolerant and some that are not. But there was a third phenotype that came out of this, and this is what they call acquired tolerance. So in this case, so these data are still coming in. This is relatively new. But one of the things is, so basically what I mean to say is I don't know the genotype of these people that I'm saying have an acquired lactose tolerance because that data is not in yet. But when it came to a blood glucose response, there was nothing, no spike. So as I just told you before, this would suggest that they are lactose intolerant, but there was no gas production either. So the big question from this is, oh, sorry, I thought I had another thing there. Where did the lactose go? And again, I don't know the answer to that at the moment. As I said, this is ongoing research, but I wanted to show you some newer unpublished results as well. So figuring this out is definitely something that many people in the lab are working on. So one of the things will be, for example, to feed C13 lactose. Ideally, this would have been nice to do in humans, but it's rather expensive to do the heavy isotope labeling with the lactose because of the quantities needed, because when humans are doing this, you're taking about 25 grams. So instead, this will be done in bioreactors. And because of the effort that it took to get these samples and these questionnaires, so many other projects will come out of this cohort as well. I realize I went through this kind of quick, but that is a good thing, so maybe we can kind of catch up a little bit. So I just want to acknowledge I work with a fantastic group of people, Director Ruth Lay, Alejandro Reyes, who is the tandem group leader as well, and of course, thank you to all of you for staying here and for your attention today. Thank you.